All right, so for the next couple of weeks, we're going to be investigating three different processes that are ways that cells can get and use energy. We're going to begin with this process of photosynthesis. It's going to get a little complicated, so bear with me, but there's going to be 2 lessons. This is the first of 2 lessons. This one on the first part of the process of photosynthesis, and then the next one on the second part that actually produces the outcome product. Now, something to bear in mind is that without photosynthesis, we would have little to no life on the planet. There are some organisms that can get energy without using the sun, but pretty much everything on the planet requires photosynthesis at some point to supply it with energy. And it's this process of photosynthesis that is able to capture the sunlight that other processes can't capture that sunlight. And that's where the majority of our energy comes from on this planet for living things. So these organisms that I'm talking about, of course, are autotrophs. Autotrophs can capture the energy from sunlight, and they use the sunlight energy to make carbohydrates. Carbohydrates is sort of the basis of all of the energy that we get, and we've already talked about that. For the most part, we're talking here about plants and about algae. We're also talking about some forms of photosynthetic bacteria, although they might use a different process than the one I'm going to talk about. But for plants and algae, this is going to take place inside the chloroplast in these structures called thylakoids. So let's take a close look at the chloroplast. This is the chloroplast organelle. Within there, you will see stacks of membrane that are called thylakoids. I kind of think of them as stacks of pancakes because I kind of think of everything as food, I think. But these are the stacks of thylakoids. Together, one stack is called a grana. And in between is fluid with enzymes, and it's called the stroma. And remember, there's 2 membranes. It's a double membrane organelle. Let's talk a little bit about the process overall. This is a whole summary of the process of photosynthesis. In this process, 6 molecules of carbon dioxide together with 12 molecules of water and the energy contained within sunlight and some enzymes in the plant cell produce 1 molecule of glucose, which becomes the plant's biomass. 6 molecules of oxygen are generated, as well as 6 molecules of water. Okay, so the net water is 6 in, because you have 6 in and, 12 in and 6 out, so the net is 6. You need water to do this thing. So we're going to talk about all the steps that produce this whole process. Let's start by talking a little bit about sunlight. Remember, photosynthesis is based on the energy in sunlight. Sunlight is also known as electromagnetic radiation. That's another word for it. And it comes from the sun, obviously. The thing about sunlight is that it behaves like a particle. We call it a photon when it's acting like a particle. It also behaves like a wave, so we can talk about its wavelength and its frequency like you would with waves. Now, when you talk about different wavelengths or frequencies of light, it's actually different colors of light. We perceive light as different colors when it has a different wavelength, or we can think of it as really different amounts of energy. So this is what we call the electromagnetic spectrum, and it represents all the different wavelengths of sunlight. In this case, all of the energy from the sun is a bunch of different wavelengths. The ones that we see, we call visible light, is in this narrow range from about 400 nanometers to about 700 nanometers in wavelength. In the high frequency but small wavelength end are the violets. Extending beyond is the ultraviolet. And then we have a whole rainbow going through. Make sure you get your rainbow colors correct. All the way up to about the 700 nanometer range, which is the reds of the longest wavelengths, and beyond that is infrared. We experience that as heat. Now, plants have these molecules called pigments that are colorful. The pigment by nature is colorful, and they absorb different wavelengths of light. So they absorb some, and we can think of that as some colors, and then they're going to reflect or transmit other colors, other wavelengths. Chlorophyll is the predominant pigment that we see in plants, and it happens to absorb all wavelengths except in sort of the yellowish greenish range. So we see plants as greenish in color because the chlorophyll, the predominant pigment is chlorophyll, and it's going to reflect that green light. That's what we see. There are actually two chlorophylls. There's chlorophyll A and chlorophyll B. They're slightly different greens. And I just wanted to show you their structure. These are the structure of the two different chlorophyll molecules. Notice it's kind of a long hydrocarbon chain here on the end. And then there's a very complex molecule on the other end with 4 nitrogens holding into place a magnesium ion. So magnesium is extremely important to plants, as you probably have heard. I don't know if you've heard, but like if you're growing plants, your soil needs magnesium in it, and that's why, because they cannot make their chlorophyll that they need without it. I wanted to show you a little bit about what's called the absorption spectrum of the different pigments that you find in plants. You can see the 2 green lines here and here. This one goes up and this one goes up over here. We have chlorophyll A and chlorophyll B. This is showing where it absorbs the best. So if you see for chlorophyll A and B, it absorbs real well in the blue-greens, and it absorbs in the reds, but down around here in the middle in the sort of the yellow-green range, it doesn't absorb at all. So you'll notice that there are these other colorful lines. We have lutein, we have beta-carotene. You may have heard of that. This is a xanthophyll. We have lycopene. You may have heard of that one. That's in tomatoes. And they absorb in slightly different wavelengths, so that they're covering the absorption of some of the light in this 500 range around in the greens. Here are some other pigments. Here's some reds and some purple pigments. And of course, most plants have more than just chlorophyll in them. They have these other pigments that can absorb light in some of the wavelengths that chlorophyll cannot absorb, so that the plant can get the maximum amount of energy from the sunlight. And so you'll see a lot of different pigments in what appears to be green leaves, and we're going to talk more about that, and we actually have a lab coming up on that. So we've talked about an overview of the whole process. We have sunlight energy being captured, and it gets captured first in a molecule of adenosine triphosphate, ATP. You may have heard of this. And ATP is the energy that cells use. So everything that the cell is doing, either breaking down glucose to get energy or capturing sunlight to get energy, it's going to be creating this molecule ATP, because that's how cells use energy. And ATP is generated from adenosine diphosphate, which has 2 phosphates, and the addition of an extra phosphate, which makes it triphosphate. And it's that phosphate bond that contains the energy. ATP is then going to be used to convert carbon dioxide into glucose in the dark reaction, or the Calvin cycle. During the reaction, water is going to be split, electrons are going to be moved around, and oxygen is given off as a waste product. So that's kind of the 3 steps that are going to happen. There are 2 main parts I keep mentioning: the light reaction and the dark reaction, also known as the light-dependent reaction, and the light-independent reaction. The light-independent reaction, or the dark reaction, is also called the Calvin cycle. So you might hear those terms coming and going. Here's another overview, and I guess I'm being redundant, but I'm afraid that it's complicated, so I want to go over it again. So here's an overview of the whole process. We have sunlight coming in. We have a process called photophosphorylation, which we're going to get into. In this process, water is going to be split and oxygen is generated. We're going to generate some ATP. We're going to generate something called NADPH. And these 2 molecules are going to be part of this Calvin cycle, or dark cycle. In this cycle, carbon dioxide is going to be converted into glucose, or sugar. So let's get into the details of the light-dependent reaction. Plant pigments, as I mentioned, the chlorophylls, are actually grouped into 200 to 300 pigment clusters. So they're grouped together, they're not by themselves. And these clusters are called photosystems. There are 2 of them, photosystem 1 and photosystem 2. We call them PS1 and PS2. And they're located embedded into the thylakoid membrane, as I mentioned before. As light hits the pigments, the energy from the sunlight make electrons boost up in energy level. So they're going to jump in energy level, and I know you know what that is because you've had chemistry. The energy then is going to be passed around from pigment to pigment, boosting up these electrons until it reaches a specific reaction center, a specific photosystem called a reaction center, and it's called P680 because it absorbs, its maximum absorption is at 680 nanometers. That's exactly the kind of pigment it is. When it's hit there, water is actually split into hydrogen and oxygen ions. This is called photolysis. Photo meaning light and lysis meaning splitting. It's splitting the water apart using the sunlight. So it looks something like this. We have a photon of light coming in, strikes a pigment molecule, boosts electrons. It's like a chain reaction until it finally hits P680. And when it hits P680, that's going to break apart this water into oxygen and hydrogen. The electron is going to be passed along, and so we're going to move on to that step. And this is at photosystem 2. Actually, photosystem 2 comes before photosystem 1. It sounds weird, but that's how it works. At this point, electrons from P680 are going to be transferred to photosystem 1, which we call P700, which is the second reaction center. P700 meaning has its maximum absorbance. That pigment has its maximum absorbance at 700 nanometers. It's going to bounce through a series of proteins that are embedded in the thylakoid membrane, and this is called an electron transport chain because the electrons are transferred between these proteins. Once it reaches P700, sunlight's going to boost the energy again. Okay, there's going to be another photon of light taken in, and this time there'll be enough energy to generate a molecule of NADPH from NADP and hydrogen ions. This is an electron carrier, they're called. NADPH is an electron carrier, and it's going to carry the electron away from the thylakoid. Did I say that right? Yes. So this is an electron carrier that's going to carry that. Very important. All right, so here's an overview of this. We have the electron transport chain. It's going to carry those electrons. Eventually, it's going to reach P700. At that point, there's going to be another photon of light bouncing around from pigment to pigment, and it's going to also reach P700. That's going to be enough energy to boost that electron high enough to convert NADP plus and hydrogen ion into NADPH, and it's going to be carrying, boom, boom, 2 electrons. So here's the whole process as one picture. We have a single photon of light, boosts electrons in energy level. It's going to eventually reach P600, boost those electrons. When that happens, water is split. Then the electrons are going to be carried from protein to protein, still embedded in the thylakoid membrane. It's going to be carried along to P700. At that point, there should be another photon of light at least, photosystem 1. It's going to boost again, and it's going to be enough energy to convert NADP into NADPH, again carrying along a couple of electrons. Remember, photosystem 2 is first, and then photosystem 1. P680 first, then P700. All right, the final step in the light reaction, or the light-dependent reaction, is to generate some ATP. As the electrons are passed along in the electron transport chain and from photosystem to photosystem, hydrogen ions are attracted, because remember, electrons are negatively charged, hydrogen ions are positively charged, and so they're attracted. They're going to follow along. They begin to build up inside the thylakoid. They do that because they're ions and they cannot cross across the membrane, but they're still attracted there, but they can't come across. This sets up a very strong hydrogen ion concentration gradient. Okay, we sometimes call these protons, by the way. It's a strong proton gradient. Then there is a specific protein channel that will allow the hydrogen ions through, and it's called ATP-Synthase. It's an enzyme, and it's also a pore. So the hydrogen ions can pass through only here at ATP-Synthase. When they pass through, they're going to catalyze a conversion of ADP to ATP in the process. So this ATP-Synthase is like a factory for making ATP. It's powered by the movement of these hydrogen ions. So let me show you what that looks like. Here is, again, the whole process. We've got our P600, the electron transport chain, P700, another bit of electron transport chain. Okay, all the while electrons are being transported, these hydrogen ions are building up. You see all these hydrogen ions inside the thylakoid. Okay, here we're making some NADPH, very important, going to the dark cycle. But here's what we're talking about here. These hydrogen ions can only come through right here at ATP-Synthase. at this pore, which is also an enzyme. So it's here that NADP plus some free phosphate is catalyzed to create ATP, and that's how the ATP is generated. Now, why is the cell doing this? It's capturing the sunlight into this molecule of ATP, and this molecule of ATP is going to be used to power the production of glucose. So that's the whole reason behind the light reaction or the light-dependent reaction. Sunlight is being captured in a molecule of ATP. Okay, if that is not confusing enough, I don't know what is. I'm going to make a second video and you'll be able to see what happens after part 2.